I am sitting here in a place which is a matter of great emotion and pride for me, I guess, in exactly the building where I was born downstairs, and now the upstairs I have developed into a research center for as long as I work and I have a working life. I would like to work here. So here you can see that we have a lot of spaces where we are bringing the rice over from the adjacent field. You see the footage of the field also. We are concentrating on several phenotypes of rice. As you know, we develop multi-harvest rice, Panchabrihi, which means that when you cut it, it grows again. You get one harvest in Boro. You cut it, it grows back in 45 days, gives a second harvest. The same plant gives then a third harvest, fourth harvest, fifth harvest. This is a phenotype. A phenotype we developed, a phenotype we increased tremendously, because there is a residual phenotype which is called ratoon. So whatever that ratoon pathway is, we have super activated it, and now there are multiple crops coming from the same plant. But we are doing it in a very, very careful and focused way. So a particular plant, we want to know what is the grain per panicle, how many panicles does it have, what is the weight of 10,000, 1,000 grains, and then finally how many plants are there in a hectare, which is 100 meter this way and 100 meter orthogonal to that. So that phenotypic characterization of every variety in the minutest detail is most important in order to understand productivity. So productivity of rice is nothing but in 100 meter by 100 meter, 10,000 square meter, also known as hectare. So whether you are putting 100,000 plant in 1 hectare, or 160,000, or 250,000, or 360,000, that density of packing is a factor in productivity. Number two is that how many grain per panicle is an inherent property of a variety. It's genetic trait that how many grain it makes per panicle, how many side panicle, and in fact this area has a unique name for the side panicle, which are called puki. So these lateral side panicles, the more the inflorescence have side panicles, the more grains they have. Finally, how many grains per panicle, and how many panicles per plant, and how many plants per hectare. These are the individual factors which in a combinatorial way determines the yield per ton. People who don't understand this individual genetic component, how it contributes, comes together in a particular variety, they don't understand the genetics of rice productivity. So however, this rice productivity phenotype can be understood in this low-tech condition. We don't have any microscope, we don't have any gel, we are not isolating any DNA. We don't have a microbiology lab, nothing. What we have is a modern thinking. What we have is a thinking around the phenotype and breaking it up into individual components, teaching students the importance of these components, showing how different stress affects these different components. And that is how we can have a good grip on the phenotypic trait of rice and ultimately predict the yield of rice. So this is what we are doing in this village for rice. And recently we brought in also corn or maize. Corn is the Z maize, is the kind of the mother of modern genetics, developed in the US, particularly where I spent long years. So I spent time in Cold Spring Harbor even when Barbara McClintock was active, and I used to see her collecting corn and for her experiment, and that triggered me into wanting to do corn. It's a kind of an imitation syndrome, I think. Barbara was doing it. Everybody said, what does Barbara say about this? She was the ultimate kind of a wise woman of genetics. So I came in contact with her, had a long discussion with her on crossing over cytogenetics in the presence of Robin Holliday. In those days, in 1984, when there was a conference called Recombination at the DNA level, I had just discovered REG-D gene, so I was invited. Frank Stahl, who recently passed away, was there. So I interacted with these people in that rarefied atmosphere, and I became a geneticist in their presence. So in that village, when I came back to Bangladesh, I was lamenting the situation that why doesn't Bangladesh have good genetics? If I go after university, there is no strong genetics curriculum in this country, which should change. So I'm telling everyone, including the current advisor of education, that please, let's change the curriculum. Let's teach people what is relevant. Let's teach people what is modern genetics, and so on. So here again, if somebody were to come here, some students were to come here and spend some time here a week, we can teach them all the genetics they need. They don't have to learn it from the textbooks. Genetics is not a field that you should learn from textbooks and counting, just looking at the picture of what Mendel did. You ought to do the exact experiment that Mendel did. So we have those pea plants, we have the corn plants, we have the rice plants. We are using it for education. We are using it for food security because the colored corn, we have pioneered the use of colored corn. Many colors we have created by combinatorial stacking of genes in a conventional genetics way. We don't do GMO, but we could create many colors, which means many small molecules which are nutraceutical, which helps you for your health. So first of all, we are doing rice genetics, targeting Panchabrihi phenotype, but other phenotype also. Colored rice, we have brought back traditional rice. Number two, in a big way, we are doing corn, and then we have just started to do hybrid wheat. I'm always interested in heterosis and hybrid wheat. So I had this ambition that I should develop hybrid wheat for Bangladesh. So that's all I wanted to say for now. Thank you.